In 1972, an evolutionary biologist at the University of Chicago named Richard Lewontin sat down with data from 17 different genetic markers, collected from human populations spanning every inhabited continent on Earth, and ran a calculation that would go on to become one of the single most cited and most fiercely argued over results in the entire history of human genetics. Lewontin divided humanity into the 7 broad racial categories commonly used at the time: Caucasian, Black African, Mongoloid, South Asian, Amerindian, Oceanian, and Australian Aboriginal, and then measured exactly how much of humanity's total genetic variation could actually be explained by which of those 7 boxes a person happened to belong to. The answer he found was, and remains, genuinely startling: less than 15%. According to Lewontin's calculation, more than 85% of all human genetic diversity exists among individuals within any single one of those racial categories, not between the categories themselves. 2 people randomly selected from the same continent, the same country, even the same village, will, on average, carry almost as much genetic difference between them as 2 people selected from opposite sides of the planet. That single number has been cited, contested, defended, and re-argued for more than 50 years now, and it sits at the center of one of the most genuinely important and most genuinely misunderstood questions modern science has ever tackled: whether human races, as most people intuitively understand the concept, actually correspond to anything real and coherent at the level of our DNA. To understand why Lewontin's finding landed with such force, and why it continues generating debate more than half a century later, you first have to understand what scientists actually mean when they talk about apportioning genetic diversity. Every human being alive carries a genome built from roughly 3 billion individual base pairs of DNA, and while the overwhelming majority of that genetic code is essentially identical across every person on the planet, regardless of ancestry, there remain millions of specific locations, scattered throughout the genome, where individuals differ from one another. Lewontin's method involved measuring how those specific points of variation were actually distributed. If human races corresponded to something genuinely deep and biologically fundamental, the kind of division you might expect between genuinely distinct subspecies, you would expect to find that most genetic variation clusters neatly between those groups, with individuals inside any given racial category looking relatively similar to each other and relatively different from people in other categories. That is not remotely what Lewontin found. Instead, he found that the genetic differences separating any 2 individuals, chosen at random from the entire human species, are overwhelmingly differences you would find between any 2 people anywhere, regardless of which continent, country, or traditional racial category either of them happen to belong to. Follow-up research conducted using far larger genetic datasets and far more sophisticated sequencing technology than existed in 1972 has repeatedly reproduced remarkably similar numbers ever since, with subsequent large-scale studies consistently finding that somewhere around 85 to 90% of measurable human genetic variation exists within traditionally defined population groups, while differences between those groups typically account for something closer to 6 to 10% of the total, figures that have proven remarkably stable across dramatically different datasets, genetic markers, and analytical techniques spanning 5 full decades of research. To fully appreciate why this scientific debate carries so much weight, it helps to understand where the concept of dividing humanity into a handful of discrete races actually came from in the first place, since the categories themselves predate genetic science entirely by roughly 2 centuries. The modern taxonomic tradition of sorting human beings into a small number of races traces back most directly to the German naturalist Johann Friedrich Blumenbach, who in the late 18th century proposed a 5-part racial classification system: Caucasian, Mongolian, Ethiopian American, and Malay, based primarily on his own subjective assessment of skull shape and physical appearance, developed decades before anyone possessed even the most rudimentary understanding of genes, DNA, or heredity at the molecular level. That 18th century framework, developed using tools and assumptions we would now recognize as scientifically primitive at best, nonetheless proved remarkably durable, shaping censuses, immigration policy, medical research, and everyday social categorization across much of the world for the following 2 centuries, largely because it matched the intuitive, surface-level visual sorting most people already did, informally based on skin color and facial features. When genetic science finally developed the tools to actually test whether those centuries-old categories corresponded to anything real at the level of heritable biology, starting with Lewontin's landmark 1972 analysis, it was, in a very real sense, testing an 18th century classification scheme against 20th century molecular evidence for the first time, and finding a considerably weaker correspondence than that classification's remarkable cultural staying power might have suggested. He drew a direct, explicit conclusion from it, stating in his original paper that human racial classification carries no genuine social value, and that, in his own words, it is positively destructive of social and human relations, concluding that race carries virtually no genetic or taxonomic significance as a meaningful biological category. That was, and remains, a genuinely bold scientific claim, and it did not go unchallenged. In 2003, the statistician and geneticist Anthony Edwards published a direct critique of Lewontin's broader conclusion, one that has come to be known, somewhat controversially, as identifying Lewontin's fallacy. Edwards did not dispute Lewontin's actual numbers. He disputed the interpretation drawn from them. His argument centered on a genuinely important statistical point that Lewontin's original 1972 analysis looked at each of those 17 genetic markers individually, one at a time, in isolation from one another, when in reality, human populations carry correlated patterns across many genetic markers simultaneously, and those correlated patterns, considered together rather than one marker at a time, can still allow a sophisticated statistical model to sort individuals into recognizable geographic clusters with genuinely high accuracy, even though no single marker on its own reliably distinguishes one group from another. That specific critique found powerful empirical support just 1 year later in a landmark 2002 study published in the journal Science by geneticist Noah Rosenberg and colleagues, examining genetic data from more than 1,000 individuals across 52 populations worldwide using a computer program called Structure, specifically designed to detect hidden patterns of genetics, clustering without being told in advance where any individual sample actually came from. When the program was asked to sort the data into a small number of genetic clusters, it produced results that correlated strongly with genuine geographic and continental ancestry, with individuals from Africa, from Europe and Central Asia, and from East Asia and the Americas generally sorting into recognizably distinct statistical groupings. An additional analysis able to separate Native American populations from East Asian ones when a slightly larger number of clusters was requested. This finding has been repeatedly and often selectively cited in the decades since as apparent scientific vindication for the idea that genetically distinct human races really do exist after all, precisely the kind of confident, oversimplified claim that later academic analysis of the paper's public reception has specifically warned against. Because here's the genuinely crucial detail that gets lost whenever this particular study gets cited to support a simple, discrete racial classification scheme. The clustering Rosenberg's team identified was not stable, and it was not naturally discrete. When researchers instructed the software to identify a different number of clusters than originally tested, the boundaries between groups shifted, sometimes dramatically, with some individuals reassigned to entirely different statistical clusters, depending purely on how many total categories the researchers asked the program to sort humanity into in the first place. A genetic clustering algorithm that produces different unstable group boundaries depending on an essentially arbitrary choice about how many total groups you decide to look for in advance is not describing a set of natural, fixed, pre-existing categories. It is describing something closer to a spectrum, a gradient of genetic similarity shaped overwhelmingly by simple geography and historical patterns of human migration, one that can be sliced into however many artificial boxes a researcher chooses to impose on it, without any of those particular boxes representing a uniquely correct, biologically privileged division. This distinction between a genuine spectrum and a set of discrete categories sits at the very heart of why modern population geneticists overwhelmingly reject traditional racial classification as a scientifically meaningful biological concept, even while fully acknowledging that genuine, measurable, geographically patterned genetic variation absolutely does exist among human populations. Human genetic diversity is real. It is measurable. It correlates, often quite strongly, with geographic ancestry, and forensic scientists and medical researchers alike make legitimate, practical use of that correlation every single day, estimating a crime scene DNA samples' likely geographic ancestry, or flagging that certain populations carry meaningfully elevated risk for specific genetic conditions, like sickle cell trait or Tay-Sachs disease. What that same body of evidence does not support is the idea that this variation naturally sorts itself into a small number of sharply bounded, internally uniform categories corresponding neatly to the traditional racial groupings most societies have historically used, categories like black, white, Asian, and so on. Human genetic variation instead follows what population geneticists call a clinal pattern, changing gradually and continuously as you move geographically across the planet, without sharp natural breaks separating one supposed race from the next, the genetic equivalent of a smooth color gradient rather than a set of discrete, clearly separated paint chips. The scale and precision of the genetic evidence available to researchers has expanded enormously since Lewontin's original 1972 analysis, which relied on just 17 protein markers examined across a comparatively modest sample of populations. The Human Genome Diversity Project, launched in the 1990s and drawing on the same broad dataset Rosenberg's team analyzed in 2002, eventually compiled genetic samples from over 1,000 individuals representing 52 populations spanning every inhabited continent. The 1000 Genomes Project, launched in 2008, went considerably further, eventually sequencing the complete genomes of more than 2,500 individuals from 26 different populations around the world, generating a genuinely enormous, publicly available data set that researchers continue mining for new insights into human genetic variation to this day. More recent efforts, including the Simons Genome Diversity Project and various national biobank initiatives launched across dozens of countries, have pushed that sample size even further into the hundreds of thousands and, in some cases, millions of individual genomes. Every one of these dramatically larger, dramatically more technologically sophisticated follow-up studies has continued reproducing broadly the same fundamental pattern Lewontin first identified using far cruder tools half a century ago, that the overwhelming majority of human genetic variation exists within traditionally defined population groups rather than between them, even as these larger datasets have also continued refining and complicating the picture in exactly the more nuanced, clinal direction the Rosenberg clustering research first suggested back in 2002. And one that consistently surprises people encountering it for the first time involves the continent of Africa specifically. And it flows directly out of one of the best established findings in the entire field of human evolutionary genetics, the out of Africa model of human origins. Modern Homo sapiens evolved in Africa roughly 300,000 years ago, and the overwhelming majority of our species' entire evolutionary history, and therefore the overwhelming majority of the genetic diversity our species has ever accumulated, took place entirely within that single continent. Sometime between roughly 50 and 70,000 years ago, a relatively small subset of that much larger, much more genetically diverse African population migrated out of Africa and eventually populated every other inhabited continent on Earth, Europe, Asia, Australia, and, considerably later, the Americas. Because that founding population leaving Africa represented only a small, non-representative sample of the full genetic diversity that already existed across the African continent at the time. Every single non-African human population alive today descends from a genetic bottleneck, carrying collectively only a fraction of the total genetic variation that remains present within Africa itself. The direct, almost paradoxical consequence of that migration history is this: there is more genetic diversity contained within the African continent alone, among African populations that never left, than exists across every single non-African population combined, spanning Europeans, East Asians, Native Americans, Pacific Islanders, and everyone else outside Africa put together. Two individuals from different ethnic groups within Africa, say a San hunter-gatherer from southern Africa and a Yoruba farmer from West Africa, can be more genetically different from each other than a Norwegian is from a Japanese person, a finding that directly and completely undermines the common, deeply intuitive assumption that treats an entire continent's worth of people as a single, relatively uniform racial category simply because of superficial similarities in skin pigmentation, while treating far more genetically similar populations elsewhere in the world as obviously naturally distinct races from one another. Skin color itself, the physical trait most commonly used throughout history as the primary visual marker for sorting people into racial categories in the first place, turns out to be a particularly poor guide to overall genetic relatedness, and understanding exactly why reveals something genuinely important about how evolution actually works. Human skin pigmentation is controlled by a relatively small number of genes, and it evolved repeatedly, independently, and relatively recently in human evolutionary history, primarily as a direct adaptation to local levels of ultraviolet radiation exposure at different latitudes, with darker skin generally providing better protection against UV damage in skin, cancer closer to the equator, and lighter skin allowing for more efficient vitamin D synthesis in regions receiving less overall sunlight throughout the year, particularly at higher latitudes further from the equator, because this particular trait responds so directly and so strongly to local environmental pressure, it can evolve and shift relatively quickly on an evolutionary timescale. And populations living at similar latitudes, even on entirely different continents with genuinely deep ancient genetic separation between them, can independently evolve broadly similar skin tones through convergent evolution, without that shared skin color reflecting any deeper, more fundamental genetic kinship between them at all. In other words, the single physical characteristic most societies have historically leaned on hardest to define and enforce racial categories turns out to be one of the least reliable indicators of genuine overall genetic relatedness anywhere in the entire human genome. a trait that evolved fast, evolved independently in multiple separate populations, and tells you comparatively little about the deeper ancestral history written into the other 99.9% of a person's DNA that has nothing whatsoever to do with pigmentation. None of this scientific nuance means genetic ancestry is somehow meaningless or untraceable, and it's worth being genuinely precise about exactly where the real, well-supported science actually lands, since oversimplifying in either direction does a disservice to what the evidence actually shows. Modern ancestry testing companies, forensic genetic laboratories, and medical researchers can, and routinely do, use carefully selected panels of genetic markers to make statistically meaningful, often remarkably accurate estimates about a person's general geographic ancestry, distinguishing, for instance, West African ancestry from East Asian ancestry, from Northern European ancestry with genuinely impressive precision in many cases. That capability is real, and it rests on the same genuine, measurable, geographically patterned genetic variation Rosenberg's clustering study identified back in 2002. What the science does not support is the further leap from statistically identifiable geographic ancestry clusters to the much older, culturally and historically constructed idea of a small, fixed number of discrete biological races, each internally uniform, each clearly and naturally bounded from the others, each corresponding neatly to socially and historically defined categories like black, white, and Asian, that different societies have drawn in different, often contradictory ways across different historical periods and different countries. The American Society of Human Genetics, the leading professional organization representing human geneticists in the United States, has issued formal public statements addressing exactly this distinction directly, explicitly noting that while genetic ancestry can meaningfully inform aspects of health, identity, and even certain forensic applications, the concept of race itself remains fundamentally a social and cultural construct rather than a natural biological or genetic category, one whose specific boundaries and definitions have shifted repeatedly throughout history in ways that track social and political circumstances far more closely than they track any underlying stable biological reality. The forensic and medical applications of modern population genetics research offer a genuinely useful, concrete illustration of exactly how this more nuanced scientific picture actually plays out in real practical use. When forensic laboratories analyze an unknown DNA sample recovered from a crime scene, they can use specific panels of ancestry-informative genetic markers to generate a statistical probability estimate regarding a suspect's likely broad geographic ancestry, information that can genuinely help direct an active police investigation. But researchers working directly in this specific field have themselves published pointed warnings about the risk of the same technology inadvertently reinforcing outdated, scientifically unsupported ideas about race, precisely because forensic ancestry testing tends to deliberately select and rely on exactly the narrow subset of highly differentiated genetic markers, including visible traits like skin, hair, and eye pigmentation that show the sharpest, most exaggerated differences between broad geographic population groups, while ignoring the vastly larger remainder of the genome where, as Lewontin originally demonstrated more than 50 years ago, the overwhelming majority of genuine human genetic variation actually resides, scattered throughout individuals, regardless of which broad geographic ancestry group they happen to belong to. The same body of research carries genuinely important practical stakes within modern medicine specifically, and getting the underlying science right matters well beyond pure academic debate. For decades, medical practice in many countries has sometimes used a patient's self-identified race as an informal proxy for underlying genetic risk factors, adjusting diagnostic thresholds or treatment recommendations for conditions ranging from kidney function to certain drug responses based partly on racial category. Population geneticists have increasingly pushed back against this practice, pointing out that self-identified race, itself a social and administrative category rather than a genetic one, often turns out to be a remarkably poor proxy for the specific genetic variants that actually drive meaningful differences in disease risk or drug metabolism between individuals. A patient's specific genetic ancestry, which can vary enormously even among people who share the same self-identified racial category, or who might be sorted into entirely different racial categories in different countries, despite carrying nearly identical ancestry, turns out to be a considerably more precise and more clinically useful variable than race itself. Sickle cell trait offers a genuinely instructive example here, since it is often popularly associated with Black or African ancestry specifically, when the underlying genetic variant that causes it actually evolved independently in multiple different populations around the world as a protective adaptation against malaria, and is found at meaningfully elevated frequencies, not just across parts of Africa, but also in parts of the Mediterranean, the Middle East, and India, regions whose populations are conventionally sorted into entirely different racial categories under most traditional classification schemes, a pattern that only makes sense once you recognize the trait tracks historical exposure to malaria rather than membership in any single coherent racial group. Lewontin's original 85% figure, repeatedly reproduced across 5 decades of subsequent research using far more sophisticated tools, Rosenberg's genuine but genuinely unstable and continuous geographic clustering patterns, the out of Africa bottleneck that leaves an entire continent carrying more internal genetic diversity than the rest of humanity combined, and the comparatively shallow, fast-evolving, and independently repeated nature of visible traits like skin pigmentation, is a picture of human genetic diversity that is considerably richer, considerably more interesting, and considerably more scientifically precise than either of the two oversimplified extremes that tend to dominate popular conversation on this topic. It is not true, as some have historically claimed, that humanity naturally sorts into a small number of a sharply distinct, internally uniform biological races the way a naturalist might sort different species of finch. But it is also not accurate to claim that human genetic ancestry is somehow entirely meaningless, untraceable, or scientifically irrelevant, since geography and migration history have genuinely, measurably shaped the distribution of human genetic variation across the planet in ways modern genetics can trace with real, often impressive precision. The actual scientific reality sits in the more complicated, more genuinely fascinating middle ground. Between those two extremes, a species whose DNA tells a continuous, flowing story of ancient migration, isolation, and eventual reconnection, shaped far more by the sheer geographic distance separating populations across tens of thousands of years of human history than by any small number of clean, natural, biologically fundamental dividing lines. When scientists actually sat down and compared the DNA of people from every corner of the globe, what they found wasn't proof that humanity naturally splits into separate, distinct races. It was proof of something considerably more remarkable, an entire species whose deepest, most fundamental genetic story is overwhelmingly one of shared, continuous, deeply interconnected ancestry, with far more binding every single human being together than has ever genuinely divided us apart at the level of our own DNA.